So I work in France at the CRCINA, which is a Cancer and Immunology Research Center, which is part of the National Institute of Health and Medical Research. And I'm going to talk about oncolytic viruses. So, how do you like this? Okay. So I have no personal conflict of interest, but the virus I will talk about at the end, still there is a patent for a company, but I'm not involved, but just for you to know. So make it really simple at first. I'm not sure you're really familiar with oncolytic viruses. So oncolytic virus, basically, an oncolytic virus is a virus that can infect specifically tumor cells and kill these cells specifically without harming the healthy cells. It's been shown also over the years that these viruses are part of the immunotherapy revolution, because when the tumor cells are killed by these types of virus, they can activate immune cells. Okay. So actually there are plenty of oncolytic viruses. So these were drawings that we got a few years ago when we did the National Science Fair in France, asking children to draw what they thought were viruses. They were pretty good actually. They were young, but they had good ideas. So these oncolytic viruses, they come in very different flavors. When you look at the virus classification, so you have all these different families of viruses that are classified based on the type of the genome they have, RNA or DNA, single-stranded, double-stranded, the shape of the capsid. And if you look at, really there are a lot of different families in which you will find these cancer-killing viruses. So in the big group of RNA viruses, there are a lot of these oncolytic viruses. I will talk today about Measles virus, but we also work on vesicular stomatitis virus. And in the DNA viruses, you have some really promising agents. The most promising actually are herpes virus, and I will start with this really quickly for the introduction, because there is a virus which has this really weird name, which is Thalidomide-Pareb-Vec. So it's known as TVEC. It's a herpes virus, so it's a DNA virus, and it was attenuated to be oncolytic, so cancer specific. So the virus was modified so genes encoding a neuroviolence factor was removed, and also another gene that was involved in virus escape from the immune system, and were added 2 copies of an immunostimulating cytokine, which is GM-CSF. So this virus was the first one to be approved by the FDA and by the European Agency 3 years ago for the treatment of unresectable metastatic melanoma. So in the phase 3 trial, it showed good results with durable response improved for patients. What was really interesting in this study was that the virus could kill the tumor lesions in which it was injected, but also tumor lesions that were at a distance from the site of injection. So it means that the virus was able to activate an immune response that was then able to find other types of tumor lesions. More recently, last year, a paper in Cell showed that this virus, so it was another clinical trial, showing that this virus was doing really good in combination with anti-PD-1. And actually what happens with oncolytic viruses, and more specifically here with TVEC, is that they are really good at changing the microenvironment, killing cells in an immunogenic way. And so this will turn cold tumors into hot tumors. And then checkpoint inhibitors will be really good at maintaining the activity of the T cell that will enter the tumors. So in the lab, we're interested in another virus, which is attenuated measles virus. Actually, it's the vaccinal strain that is used for vaccination of children. There are other labs using it in the US. So they use the Edmonton strain, we use the Schwarz strain, but they are really close and derived from the same strain originally. And so we've been interested in trying to find how this virus can be specific to tumor cells and how it kills tumor cells to be immunogenic. So this is just an example of how it works. Yeah, so this is a video of mesothelioma cells that were derived from a patient. 2D culture system with a virus encoding a green fluorescent protein. And you can see with the timeline here in days, hours, and minutes that the virus spreads pretty quickly and form these syncytia that are fused infected cells. And ultimately, after 48 to 72 hours, all the cells will be killed by the virus. You can see here the lysis of the cells. So we did this. We published a paper when we screened 22 cell lines, trying to show if they were sensitive or not. Actually, we found that out of 22 cell lines, 15 were sensitive and 7 were resistant to measles infection. Then we did some experiment in 3D spheroid system. So these are cells from another patient, and we created these spheroids and we infected with a virus, and we showed that the virus was able to enter the spheroid and kill all tumor cells. Obviously, when you try to work on oncolytic viruses, you have to show that this virus won't kill normal cells. So we took mesothelial cells, and we did the same experiment in 2D with the measles virus, and you will see that few cells will be infected, 1, 2, 3 cells, but the virus will never spread in the culture. So you will have green cells in this area, if I remember correctly. So we are now one day after infection with the virus. The dose is 1 virus per cell. And still we only have 1 cell infected here, up there, and I think a second one will come here. But you won't have spread of the virus in the culture, showing that the virus is really specific for cancer cells. So in the lab, we've been also interested in trying to find if this immunogenic cell death here is able to activate immune cells, human immune cells. So I show you just one picture that we had. It's confocal macroscopy where we had these human dendritic cells that were able to engulf mesothelioma cells infected by measles virus, and we showed in the paper that these dendritic cells were then really good at activating specifically T cells that could recognize specific mesothelioma antigens. So showed you that TVEC went to phase 3 trial and went to the clinic. For measles virus, it's a little earlier actually. At Mayo Clinic, there is a lab, they are more advanced on working on this. So they had this pilot study in 2 patients that had multiple myeloma resistant to any therapy. And so it was published in 2014. These patients, they injected them with measles virus in the bloodstream, and they were analyzing lesions on the skull of multiple myeloma. And after treatment, the lesion disappeared, and they showed that there was a viral replication in this lesion. They did also a different phase 1 trial on ovarian cancer with intraperitoneal injection of the virus. And I don't know if it's still open, but they have a study on mesothelioma actually with intraperitoneal injection, intrapleural injection of the virus too. So how does the virus specifically infect tumor cells? So the wild-type virus, measles virus, has 2 receptors on cells. It's CD150, which is found on lymphocytes, and that's what makes the original wild-type virus specific for immune cells. And there is another receptor which is called Nectin-4, but when the virus was attenuated in the 50s and the 60s to create vaccines, it acquired the ability to target another receptor, which is CD46. And CD46 is found on all cells in the human body at a basal level. Actually, it's a protein that protects cells against the complement, so the cells won't be killed by the complement around when there is an immune response in the tissue. What's interesting for us is that CD46 is really overexpressed in almost all human cancers, so especially mesothelioma, but other cancers. And so you will have more CD46 on the surface of all these tumors, and so more CD46 you expect to have more virus to enter and to replicate, and you have less in healthy cells. So that was what was believed for a long time because it explained a lot. But when we looked in our experiment, actually we found some mesothelioma cells that had high overexpression of CD46 and no viral replication, and the reverse, low CD46 and high replication of the virus. So there was something else going on. So that's a work that was performed by Jean-François Fontenot, a senior researcher in the lab with a PhD student, Tiffaine Delaunay. And when we screened our MV-resistant or MV-sensitive cells, we found that there were some deletions at the genomic level. We first saw that there was deficiencies in the type 1 interferon response. So, as you know, the type 1 interferon response is really important to fight viral infection. So you have interferon alpha and beta. And so you can see that NV-sensitive cell lines really often lose both copies of the gene encoding interferon beta. So when we look at the location of these genes, what's really interesting, it's on chromosome 9. And on chromosome 9 you have CDKN2A, so P16. You have, was removed actually, but MTPA. And so you have the type 1 interferons here on the left. So it's really close to P16. And when you look at the copy numbers, also you have 0, 1, 2, 0, 1, 2 for each cell line. You can see that, for example, for this cell line, there is loss of the whole chromosome area. So the P16 gene are lost, and the type 1 interferon genes are lost too. So it's known that P16 is loss of P16 is really important in mesothelioma, but maybe the loss of type 1 interferon is also important because it's important in the anti-tumor immune response. And we had one cell line, actually, Meso 225, that had lost type 1 interferon genes, but not the P16 genes. So it may be also important for the progression of mesothelioma. Anyway, this is important for oncolytic virotherapy because if you lose the type 1 interferon genes, you're more sensitive to viral infection, and that was showed for other oncolytic viruses, including TVEC. So with this, I will talk about a research project that we had in the last few years, working with my PhD student, Joëlle Nader. She's PhD now. We thought that even if the measles virus was pretty good at killing mesothelioma cells, maybe we could improve it a little and try to maybe kill faster or kill in another way these mesothelioma cells. So this here is the measles virus genome. It consists of 6 genes encoding 8 proteins. You can see that the P genes, the second one, encodes 3 proteins, 2 of them being non-structural protein, C and V. These are the proteins that are responsible in the wild-type strain for the virulence. Of course, they've been attenuated with the passaging for vaccination. And the C protein is really interesting. It had a lot of different functions. Obviously, when you have only 6 genes for a virus, you need your proteins to have a lot of functions. C protein is important in antagonizing the type 1 interferon response, but also in inhibiting apoptosis and acting as a processivity factor, meaning that it helps the polymerase staying on the RNA strand to replicate the whole genome without getting off. So what interested us in this protein is this ability to inhibit apoptosis. So it means that in the virus, the C protein will retain this activation of apoptosis, so the virus will be able to replicate longer and then have a better progeny. Still, we thought that maybe if we remove this protein, we could have faster induction of cell death, and that would be really good for oncolytic activity because you could kill more tumor cells quickly, and that can be important. So we checked this in vitro. We had measles virus and measles virus delta C, so the C protein both encoding GFP. These are normal mesothelial cells. So we showed that as for the parental MV, the delta C was not able to efficiently infect healthy cells. So that was good. Retained the specificity for tumor cells, and actually looked maybe better, but cannot confirm. And when you look at mesothelioma cells, we had really a difference in profile. So the measles virus, the classical one, was really good for infecting these 3 cell lines. We did 12 total, but it was the same profile. So you can see that infection is taking really importantly, and so you have this fusion, characteristic fusion of infected cells. So you see a lot of green, a lot of production of the viral protein. At first we saw this and we thought, okay, delta C is not really good because you don't see that much green, so is it really infected or not? Actually, you can find green, it's just less green, but what's important is that it's killing really fast. If you look at Mesos 13 here, it's clearer. You can see at the end that there are no cells remaining, okay? So it's killing really, really quick. We checked this with another staining, so we used now the virus is in red, and we used a probe to find for activated caspase-3, which is a good marker for apoptosis. And we can see that the activation of cell death is really earlier compared to the normal measles virus. So normal measles virus will be better to replicate a lot and have a lot of viral proteins produced, while delta C will have less titers of virus produced, but will kill faster. So is this good or not? We wanted to check in vivo. It's not easy to work on measles virus in vivo. Of course, we're really interested in immunotherapy, and still we cannot work in immunocompetent models because measles virus cannot infect murine cells. So you have to work at first with human cancers, with xenografts in not-skin mice. So that's what we did. We still retain the IP challenge and IP treatment, just to be orthotopic, even if it's with 2 species. So what we found is that MV delta C was at least as good as MV to induce tumor regression. So the tumors were allowed to grow for 3 weeks intraperitoneally, and then we injected the virus and checked 2 weeks later. We recovered the tumors and we just waited them. So the virus is as good as an oncolytic. Then we're interested in trying to find if the cell death that was induced by the virus was immunogenic or not. So we first focused on the cell-derived molecule. So we looked at HMGB1. HMGB1 is a protein that is supposed to be found in the nucleus. And upon immunogenic cell death, it will be released in the environment and will be recognized by immune cells. If you have a clean apoptosis, it will be retained in cells, and so you won't have any activation of the immune system. What we found is that when we used MV delta C, we always had a lot more HMGB1 in the environment than with normal measles virus. On the other side, we were also interested in finding if there were more danger signals coming from the virus. So as I told you, the C protein is really important in helping the polymerase to do a good job. And when the C protein is not here, the polymerase will get off the strand and start again, and so on, and will create the double-stranded RNA structures that are really good inducers of immune activation, especially by recognition from the toll-like receptor 3. So we did a staining in our mesothelioma cells, trying to find if we had more double-stranded RNA in these cells, and actually that's what we found. So with measles virus, parental measles, I don't know if you see really well the fluorescence. It's really good here, actually. You can't see a lot of red here with the measles virus infection, but with delta C you will have the cells are completely red, and so you have a lot of double-stranded RNA, and this double-stranded RNA will be released upon cell lysis and will be maybe able to activate immune cells. So that's what we looked right after with another student who is just starting his PhD with me, Tatien Petithomme. So we took monocytes from healthy donors and we differentiate them in dendritic cells for 5 days with IL-4 and GM-CSF, and wanted to check if these dendritic cells will activate upon a co-culture with these infected mesothelioma cells. So we looked at the marker CD83, which is found on the surface when the dendritic cells go to maturation. So you have here the two negative and positive controls of maturation. And so you can see that delta C is by itself is able to activate a little these dendritic cells. And what's interesting is here, when you take the mesothelioma cells, non-infected or killed by UV irradiation, which is known to be non-immunogenic, you do not have activation of dendritic cells. But when these cells are previously infected with measles virus, or even better with delta C, there will be a strong activation of the human dendritic cells. Conclusion for this new virus is that has promising oncolytic properties against human MPM, and that should be something considered for the treatment of MPM. Maybe not measles virus, maybe other types of viruses, but with the TVEC going to the clinic and all this, we think that these agents can be really good to really debulk the tumor first. Second, you can activate the immune system really efficiently. And oncolytic viruses are interesting because they can be modified. You can add several genes inside, and so based on the advances in science, maybe you can find a new protein, you can put an antibody in it, and this will be produced directly in the tumor microenvironments. So that could be really interesting, especially when you have agents that are toxic systematically. This new virus is good because it has a rapid tumor cell death induction, it has an improved immunogenicity, even if we still need to characterize better in immunocompetent models. That's not easy. We just created some murine tumor cell lines that can be now infected with measles virus, so we are trying to develop this project in vivo and maybe try combination, obviously, with checkpoint inhibitors and other types of therapies. So this is summary. Now I like this picture because this was done for TVEC. It was published by another group two years ago. It shows how complicated things are now with also immunotherapies, but with viruses maybe it's a little more complicated. So you have here the TVEC virus that will enter. So this is a tumor cell, this is a dendritic cell, and this is a T cell. So TVEC will enter in tumor cells and will replicate in the cells because you will have several alterations in tumor cells that allow replication of viruses. The most easier to understand is this loss of the type 1 interferon response, but you can have other types of pathways that are mutated. So the virus will propagate, will here in the case of TVEC will produce GM-CSF, and will induce cell lysis, and you will have production, release of viral and tumor antigens. There will be also production of DEMPs and PEMPs, so immunogenic molecules that are derived from the cells or from the viruses. And all these signals will be recognized by dendritic cells, and dendritic cells will integrate the signal. They will be really activated through the TLRs. They will present the tumor antigens on the surface and activate T cells. So oncolytic viruses is like exactly the type of immunotherapy you want. So then it has to work, but that's why it could be really good in combination with checkpoint inhibitors, because it will wake up the immune system, and then checkpoint inhibitors allow to just modulate exactly what we want and maintain the response over time. So with this, I will thank my team, obviously, Marc Grégoire, who is the leader of the lab, other people working on the virus. We also have people in the lab working on new epigenetic drugs to activate the immune system. We have something about nanoparticle delivery also. And our collaborators at the Pasteur Institute, who actually did the virus, and funding agencies, that was mostly public funding, and patient association. And thank you for your attention.